our body, the inactivated X is the recessive mutant form that has no effect on the fur. It's a mutated gene whose enzyme is inactive and cannot produce the orange pigment. On the black splotches, now there's another gene here we'll talk about in a second. This is a gene that produces black fur. And you notice, well, I'll stick on this one, so the orange patch here. So this is an autosome, separate chromosome. I don't know how many chromosomes are in cats, so I can't say. But at this particular locus, here's a gene that influences the amount of melanin produced. And we see it's homozygous dominant. So this would lead to black fur. But if the orange allele is active, it masks the expression of this gene. What do we call when one gene masks another? What's that genetic phenomenon called? Talked about it in chapter 12. Starts with an E. Epistasis. So this is epistasis. When this gene is working, it suppresses the expression of this gene here. Okay? All right. Now, in the black regions of the animal, we see now that the X chromosome with the functioning dominant allele for orange fur color is now that's now compressed into a bar body because of the random selection of this X chromosome, the expression of the ZIS gene to code itself, become inactive, and the functioning X chromosome now has the mutant allele, which doesn't work. So there's no orange fur produced, and that releases the epistatic inhibition of the B gene so it can produce the black fur on the autosome. So when you see these kind of splotchy coat colors in mammals, that's an indication that this might be due to an X-linked gene, and it's being expressed in sort of a patchy pattern because of this X inactivation, of a random selection of an X chromosome in different populations of cells in the female. Now that begins again in an early embryo stage, okay? But when a female matures and when a female makes eggs, this inactivation is eliminated. So early in the germline, both X's are functioning. Okay, it's only at about this 100 cell stage does the cells begin to choose one X over the other. So prior to that, it's kind of, when you start to make eggs later, you kind of reset, kind of remove that inhibition, and both X's are available for your offspring. Someone had a hand up somewhere? So it cycles. It goes inactivated and released. Okay. Interesting phenomenon. Does that make sense?
So at the 100, it picks which X to use, and
yeah,
then that's...
so you're one cell, a zygote, then you're 2 cells in 24 hours, then 24 hours later you're 4 cells, then you're 8 cells, and as these cells divide, they divide so fast the cells get smaller and smaller in your ball of cells. At about 100 cell stage in a female, each cell starts to turn one X off, and another cell turns another X off, and it does it at random, so they're not all the same X's. And every descendant of that cell through mitosis will keep that same X turned off. It'll never change. So a woman is a true chromosomal mosaic, okay, where we men are not. Okay. And this is evidence. This is the phenotypic evidence of that sort of random selection of X chromosomes to inactivate. Yeah.
If it's random, right? Which one is?
Yeah.
So wouldn't it not look like that if it was random? Because it kind of seems like the orange is like there's more orange in one region, and then there's a lot of black together. Wouldn't it be a complete like?
I don't know if there's one more. I mean, it's random. I mean, as far as scientists can tell, from what I've read in recent articles about this, they still don't understand how the selection is made for which X. That's all I can tell you. Okay. Now, the Y chromosome. So originally X and Y were autosomes. They were homologous. Okay, and then they've diverged over evolutionary time. And the X, again, is the most gene-rich chromosome in the human genome. The Y is the most gene-poor chromosome. The Y chromosome has somewhere around 100 genes on it. No other chromosome has as few. Okay. And there's a few critical ones here in terms of maleness. So this chromosome carries the male switch because the default development of a mammal is female. But if you have a gene on the Y chromosome called the SRY gene, stands for sex determining region of Y. Around week 6 or 7, this gene turns on, it's expressed into protein, and that enzyme starts to produce a hormone called testosterone. So this triggers testosterone production around week 6 to 7, right before it's a fetus. Still, it's a late stage embryo. And that starts to masculinize all the plumbing. Prior to that, there's a set of genetic plumbing. There's structures and tubing that will develop either into the female reproductive structures or the male structures. Okay? Like the testis in a male are really derived from the same structures as the ovaries of a female. It's the same structures. Okay, that's where the gametes are made. Okay, the oviduct in a female is the vas deferens in the male. It's just the fate of this hormone, because of this gene activated at week 6, masculinizes all those structures and turns you into a male. And that gene is right here, this tip here. Okay. This is a gene also sometimes called, this says, in this slide it says AZF. This is also known as GAZ, D-A-Z. This is a gene that influences triggers spermatogenesis. That's another male-specific gene. A number of other these are, some of these are housekeeping genes just involved in cell functioning, okay? And a few of them, actually near this short arm here, actually are homologous, have homologous counterparts on the X. So when X and Y form tetrads, the tips do recombine, but the majority of the chromosomes don't. This one's an interesting one here. This is amelogenin, this tooth enamel gene. Amelogenin is found on both, this is one of the few genes found on both X and Y, and it contributes to it produces a protein as part of tooth enamel. Okay? And actually, there's a non-coding region of this gene that varies between males and females. And in human DNA fingerprinting, this is the gene used to distinguish male from a female. So in a lot of forensic cases, you get a mixed blood sample because there's been an assailant, a rapist, a murderer. You've got victim and perpetrator's blood mixed together, and you can fish out the male and female DNA because of the difference of the non-coding region of this gene, which is on both X and Y. So you can partition the two. Kind of an interesting aside there. The fact that most of the Y chromosome does not recombine in mitosis because it's not homologous to the X means that men pass their Y chromosome intact onto their sons, grandsons, great-grandsons, and on. So all the men in this room, your Y chromosome is almost completely intact. It's the same exact as your father, his father, all the way back. And that allows people to track deep ancestry in the male lineage. Okay, if you try to track ancestry using autosomes, because they recombine at every meiosis one, every generation, as you make new germ cells, the maternal paternal autosomes recombine. It mixes things up, and it's hard to track an ancestry very deep into the past using a chromosome 1 through 22. But the Y chromosome, you can track the male lineage deep into the past to many, many generations. Okay, I'll talk about some of the exciting forensic studies. For example, they have described the Y chromosome of Genghis Khan. Okay, and also of some of the early Hebrew priests, that Y chromosome is known because it's been passed intact through the Hebrew tribes had the upper priest caste. They were called the Kohanim. That family is known as the Cohens. That's a very ancient Hebrew name, and most males of that lineage have the same Y chromosome. They dates all the way back to maybe some of the first Hebrews written about in the early books of the Old Testament. That's intact. So the Y chromosome is inherited intact generation to generation, but autosomes are not because they're recombined in prophase 1. We'll come back to that in a little bit near the end of this chapter. This is 13. We'll jump to the next chapter. Okay. Now I'm going to go real fast through this part because we've really talked about this in lab. But if you're one important difference between studying human genetics and genetics of other organisms is if you study genetics in fruit flies or pea plants or bacteria, you can perform crosses. There's no ethical issues there, but that's not really ethical in humans. Scientists don't have the ethical or the power to force matings between people because they want to observe their offspring and grand offspring. So instead, we track traits through generations, we do pedigree analysis. This is Dr. Nancy Wexler of Columbia University in New York City, studying extensive pedigrees of families that had Huntington's disease. A neurodegenerative disease is a autosomal dominant disease for which there is no cure. It leads to a degeneration of the nervous system in middle age. One of the more famous individuals I know that had the disease was a great folk singer of the Dust Bowl era called Woody Guthrie. He had Huntington's disease and died in his early 40s, mid 60s. Dr. Wexler and her group, the purpose of studying this sort of passage of this disease trait through families was to try to locate the position of the gene to understand the molecular significance of that disease. And she was able to find that the gene for Huntington's disease was on chromosome 4. And what triggers Huntington's disease is it's a disease triggered by a trinucleotide repeat. Okay? So there are parts of the genome that are incredibly repetitive. We think of a gene as being a unique nucleotide spelling, which they are, but when people have characterized the human genome, greater than 50% of all your DNA, folks, occurs more than once in your DNA. They're known as repetitive sequences. Okay, more than 50%. Right? These are sequences that occur over and over and over. Huntington's disease is triggered by what's called a trinucleotide repeat. And here are the letters, CAG. And this repeats over and over and over. Okay? And the problem is that when you have a repetitive sequence like this can lead to the number of repeats can vary easily due to replication slippage.